Welcome everybody to our weekly journal club, EV Journal Club, in collaboration with Dr. Millie Lab and National University of Singapore and Socrates Singapore. This is actually the first journal club of 2025, so happy new year to everybody. And I believe the best way to start our season of journal club is by touching a very important topic, which is gaining a lot of attention in the recent year, is the biomolecular corona. Is that kind of a layer of molecule that surround the extracellular vesicle and of course impact the normal biology of the EV. Today we have Angelo Musicò, University of Brescia, from the group of Annalisa Redghieri and Paolo Borghese. He's recently published a paper where he described a new methodology to study the EV corona. So a methodology that supposed to be less impactful on the EV structure and EV biology. So we are happy to learn more from you. Thank you, Angelo, for being here, and please go ahead.
Thank you, Marco, for the presentation. And hello, everybody, and good afternoon or good morning. I am happy to be here to have the opportunity to present our latest work about the study of the biomolecular corona by fluorescence correlation spectroscopy. So, as you know, extracellular vesicles are extracellular nanoparticles made by sulfur cells, which are involved in cell communication, and so they are involved in different physiological and pathological processes. And for cell communication, they transport and protect different types of cargoes, such as RNAs, proteins, or metabolites, and being made by cells for navigate through the body to carry informations. They are so cell-tailored for circulation and also for targeting. For these reasons, extracellular vesicles gain a lot of interest in the field in different fields of nanomedicine, such as diagnostic, due to the information they carry, and so they are interesting for example, for liquid biopsy. They are also very studied in cell-free therapy due to their low immunogenicity and their abilities in tissue regeneration. And furthermore, they are very interesting also in the field of drug delivery due to their immune evasion abilities, the targeting abilities, and the ability of carrying intracellular delivery. However, despite the several studies about application of EVs in nanomedicine, actually at the moment the lack of knowledge about their physical chemical properties is severely hampering their translation to clinic. And their physical chemical properties are, for example, in the biomolecular corona, which is gaining a lot 3 here. It's fine. Okay. Okay. I was saying, yeah, extracellular vesicles are so becoming very interesting in the field of nanomedicine, and they are studying diagnostic because they carry information, so they can give us information about the cell of origin, and so are interesting for liquid biopsy. They are also studying for cell-free therapy due to their low immunogenicity and abilities in tissue regeneration. And also they are very interesting for drug delivery, development of new drug delivery systems due to their immune evasion abilities, the targeting, and also the cellular delivery. However, their actual translation to clinics now is hampered by the lack of knowledge about their physical chemical properties, such as, for example, for the formation of the biomolecular corona, which is a mesoscale phenomenon caused by the extracellular vesicles eye surface energy due to the eye surface to volume ratio. And the molecular corona is a mesoscale phenomenon that occurs by the desorption of multiple layers of molecules with different type of bonds, stronger or softer, that are the so-called hard or soft corona. And the formation of this biomolecular corona caused the variation of the physical chemical features of the nanoparticle surface and in turn the biological identity. Biomolecular corona is a mesoscale phenomenon that has been discovered almost 20 years ago in the field of synthetic nanoparticles, and it has been acknowledged only in recent years also for EVs, where it has been found that to be functional and to have a severe impact in cellular uptake by distribution, angiogenesis, and regeneration, for example. For this reason, in a previous work with my group in Brescia, we first studied the impact of the memory corona during AV surface engineering, which is a strategy that is commonly used, for example, to enhance the targeting of EVs for drug delivery. In this work, we used red blood cell-derived extracellular vesicles, and to study the impact of the biomolecular corona, we engineered our extracellular vesicles with a fluorescent antibody, specifically the cetuximab that we used to target triple-negative breast cancer cells. And we used this antibody to engineer our EVs following two different strategies. One, chemical, where we attach covalently the antibody to the vesicle surface, and the other physical, where we let the antibody physisorbed as a corona on the vesicle surface. And then we study the functionality of these two different engineered EVs. To study the functionality, we first study on-chip functionality and in vitro. First, starting with the on-chip functionality, we perform a sulfur plasmon resonance by sensing, testing the affinity of the two engineered EVs against their target receptor. And interestingly, we found that the two different engineered EVs have the same affinity for the receptor, indicating that the strategies doesn't affect the affinity of the engineered particles. However, moving to in vitro functionality, so evaluating the uptake against the triple-negative breast cancer model, we observe very interestingly that testing the uptake in presence or in absence of FPS, so in presence or in absence of an high concentration of soluble proteins and molecules, we have different behavior. Specifically, in absence of FBS, we observe that both engineered EVs have an increase in the uptake compared to the pristine ones, while in presence of FBS, we observe that only the chemically engineered EVs still have an higher uptake compared to the pristine and the physical engineered EVs, while the physical engineered EVs uptake dramatically decrease, reaching the point of the pristine ones, suggesting a loss of functionality. To explain this, our hypothesis was that due to the presence of FBS and the high concentration of soluble molecules, this lead to an exchange on of the antibody on the AV surface, leading so to the loss of functionality and in turn the decrease in the uptake, while this didn't happen for the chemical absorbed EVs because the bonding is much more stable. So also the absorbing of particles for forming the biomolecular corona don't affect so much the uptake of DVs. However, proving this hypothesis is complicated because at this moment, state of the art about studying the biomolecular corona rely strongly on ex-situ approaches, so studying the biomolecular corona formation after the separation of DVs. But this is a problem studying the corona because biomolecular corona is actually a dynamic phenomenon which physics of molecules are constantly in equilibrium with the environment. So changing the environment, it also changed the corona. Indeed, this finds support also in literature where there are some papers that demonstrated how different separation techniques lead to the formation of different biomolecular corona that also have in turn different AV function than so the biomolecular corona affect the function of the Vs, the biological properties, but also separation techniques affect the biomolecular corona formation. So for this reason, we need to develop new in situ approaches able to study the biomolecular corona dynamic actually in their environment without separating the Vs from the environment, because it then lead to some modification of the corona. So we have not the full information. For this reason, during my PhD, I moved to San Sebastian for 9 months in the group in Spain, in the group led by Dr. Sergio Moya, in which they are expert in fluorescence correlation spectroscopy. That is a spectroscopic technique that has been used for studying the corona, also in synthetic nanoparticles. This technique is based on the use of confocal microscope, and specifically by FCS measure the fluctuation of fluorescence intensity in a small confocal volume per time, building so a autocorrelation function similarly to TLS, for example. And by FCS is so possible to resolve the composition of fluorescent objects in solution, determining the diffusion that is correlated with the size of the objects and the concentration of the different objects in solution. So we decided to apply this technique to our EVs physisolved with the fluorescent antibody, and we evaluated by FCS. We measured our EVs before and after spiking them with different concentrations of plasma proteins, so mimicking the physiological conditions. In this work, we used our fluorescent antibody, and we studied the biomolecular formation on two different extracellular vesicle subpopulation, specifically the rebel cell-derived EVs and mesenchymal stromal cell EVs, which are two different EV subpopulation that are very interesting in general for drug delivery or for drug delivery. And so we measure them before and after spiking them with plasma proteins. Before spiking with plasma proteins, we observed that the autocorrelation function of our physisolved EVs shift to the right compared to the autocorrelation function of the free antibody and the fluorophore. And by the fitting and analysis of the EV autocorrelation functions, we determine their hydrodynamic starting the hydrodynamic diameter, which is similar in both EV subpopulation, and the composition in fluorescent objects in our solution. And interestingly, in this case, measuring the composition, we observe differences between the two engineered EVs. Specifically, we observed a larger fraction of free antibody in the rebel cell-derived EVs compared to the mesenchymal ones, indicating that the antibody is weakly associated to the Rev surface, so the C2 in this case have a lower affinity for the rebel cell-derived EV surface. So we see a first sign of a compositional effect of the V membrane for the objects that are absorbed on it. So then we moved to spiking plasma proteins, and first to verify our previous hypothesis for the previous work, we measured the detachment of the antibody from the vesicle surface. And as you can see in the graph, we observed that increasing the concentration of plasma protein in both EV subpopulation, we can observe an increase in the detachment of the antibody, but following two different dynamics. So confirming again that the antibody, and in this case also the plasma components, have different affinities for the two different EV surface. And interestingly, again, we confirmed that the antibody is weakly associated with the REVs compared to the MEVs. Then, instead measuring actually the biomolecular corona formation, we evaluated the fluctuation in the hydrodynamic diameter following the spiking of plasma proteins in the two UV subpopulations. And again, in the case of REVs and MEVs, we observe two different dynamics. Indeed, in the case of REVs, we can observe that we reach a plateau in the increasing of the hydrodynamic diameter, while for MEVs, no, despite we reached a very similar hydrodynamic diameter at the higher concentration, indicating again that a biomolecular corona is forming because the hydrodynamic diameter increase due to the absorption of biomolecules on the surface of the Vs, but following two different dynamics. And this is the first demonstration that actually two V subpopulation could have also a different corona dynamics formation. Furthermore, we tried to describe better the corona that was forming on our AVs, and to do this, we applied some stoichiometric calculation based on the protein concentration we spiked and the size, average size of these proteins, and the V concentration and size. Applying this stoichiometric calculation and modeling RAVs considering the formation of biomolecular corona layer by layer. So a second layer is forming after the completing of the first layer. We determined a nominal hydrodynamic diameter that we then compared with the experimental one, the one we measured. And interestingly, we found discrepancies between these two numbers, discrepancies that correspond around the formation of one or two layers more. And also we observed these discrepancies in all the conditions we tested for both AV subtypes with similar discrepancies in numbers. And this made us try to guess the architecture of this corona that while this calculation can be only by the formation of discrete island of protein aggregates, which can increase the hydrodynamic diameter much more, and with also the formation of some patchy monolayers. And interestingly, we found also that this observation is also found also supported by the literature. For example, in this work from the group of Edith Buzas in 2021, they observed that in plasma by single particle microscopy, they observed that in plasma it's possible to observe EVs with actually this type of corona. Of course, this is a single particle measurement, while our observation by stoichiometric calculation is made in considering in bulk. So it is the average architecture of the corona forming. So, in conclusion, in this work, we developed a transferable FCS-based methodology to monitor biomolecular corona formation on extracellular vesicles. We observed that biomolecular formation and evolution could be influenced by the V origin, highlighting in this way an additional level of heterogeneity among EVs. And also we demonstrated that biomolecular corona seems to be consistent among the different EV subtypes studied, despite they showing a different dynamic in the corona formation. So I would like to thank all the people involved in this project, all the collaborators, and of course the group Brescia, and thank all of you for your kind attention.
Thank you very much, Angelo, for your presentation. Very, very interesting study. So we will have a little bit of Q&A now. So probably we can start from our audience. If there is anybody that want to ask to Angelo, please go ahead. Okay, so probably I can start first. So I will start actually from your last slide, where you say that basically the corona didn't is more or less the same between different subtype of EV, but the dynamic of the formation change. So I
Sì.
suppose the dynamic of the formation change because the abundance of the protein on the surface of the V is different among subtype. Once on the EV, you have the first layers, then the charge from the first layer of corona is the same among different subtype of EV.
Yeah.
Agree with me?
Yeah, it could be an explanation actually. Yeah, because actually, as we said in the introduction, also the biomedical corona changed the EV surface and identity, and so it could be a sort of normalization, depending, of course, on the if the because we don't have any clue about it about this type of disease is if also influence the type of proteins that are absorbing. But yeah, of course, assuming that the type of proteins that are absorbing are the same but with different dynamics, of course, then we have a normalization of the a sort of normalization of the surface that, of course, then could be an explanation about the similar architecture that we observed. Yeah.
Yeah, that's why I think it would be actually interesting to have a characterization of the EV to see the abundance of specific protein can actually affect and drive the formation of the corona.
You mean EV protein, actually?
Yes, how the abundance of specific protein can affect the formation dynamic of the corona.
Yeah, yeah. Indeed, we are starting also to perform some mass spectroscopy experiment to try also to identify which are the ingredient of the EVs that determine then the, their properties in corona formation because yes, it's interesting because yeah, we observe that different TV subtypes have different dynamics, but we don't know why, which are the player involved in this dynamic formation. So we need to explore this because yeah, could be very interesting also then once understood this also to design the EVs for drug delivery, for example, for enrich the corona in specific markers to obtain a specific after effect then.
Yes. Anybody from the audience? Okay, I will go ahead. So you start basically checking the interaction of a plasma protein on two group of EV.
Sì,
So do you believe, I believe this kind of technique can be also translated in different kind of environment.
yeah, of course.
I suppose you use red blood cells EV and stem cells because are very popular either for therapeutic application or
Exactly. We started from this because we were starting studying our EVs for developing a drug delivery system, actually. So we used to EV subpopulation that are interesting in that field. Yeah.
So I have actually another question coming from, let's say that people use a lot of EV standard.
Sì.
So it will be interesting to see the quality of this EV among them. You know, in the EV field, it's very important to standardize protocol, but I believe also it's important to standardize the material that we use to do research. So if different group use a RBCV, but the purification is different, probably also the corona is going to be different, and this is going to even make more complicated standardization across lab.
Yeah, sure. No,
So
this is a good point actually, because also yeah for yeah I start saying that the clinical translation for of extracellular vesicles is also hampered by the lack of knowledge on the physical chemical properties, and in turn they are really also connected with the aspects of the standardization because yeah, of course, knowing also physical chemical properties of EVs, we could also understand how they change between protocols of separation, of course, and yeah, because for clinical translation also we need a better standardization of the material we are using, of course.
Okay, so I have one last question. It's just a more general one. So which one
Mhm.
is the next step in your research?
Yeah, the next step in this aspect of the biomolecular corona are to applying the same technique or similar technique of studying in situ. This technique is interesting because is transferable indeed, and we can modify the subject of studying. I mean, now we studied and we characterized the interaction with using an antibody as a corona probe, but we can change molecule protein. We can study the interaction with these with specific plasma components. Um, for example, lipoproteins that are uh, in recent years are becoming interesting, the interaction with the vis as biomolecular corona. We already started to work on this, applying always FCS. And yeah, this is those are the next steps about this research in biomolecular corona.
Okay, thank you very much. I think many groups around the world are focusing their attention on the corona, AV corona for different
Mm.
kind of application, not only for the biology itself, but also the impact for diagnostic and for therapeutic, how we can
Yeah.
actually make use of the corona for therapeutic application.
Yeah.
So if there is no any question from the audience, no, thank you again, Angelo, for your time and for your
Thank you, Marco. Thank you,
presentation.
everybody, for
And
listening.
I will see you next week with the next Journal Club. Thank you again. See you next week. Bye bye.
Thank you.